Very good morning, one and all. Very good morning to one and all present on the Zoom, YouTube, and Facebook platforms. This event is organized by Center for Advanced Agriculture and Technology for Climate Smart Agriculture and Water Management, Mahatma Phule Krishi Vidyapit, Rahuri. It gives me immense pleasure to welcome you all for the international webinar series on different platforms. Under this event, today's lecture is climate resilient crops for food and nutrition security. We feel honored to have with us respected Professor P.K. Gupta sir and Dr. K.V. Prabhu sir. It gives privilege to us that our Honorable Vice Chancellor Dr. K.P. Vishwanatha sir as a chairman of today's event. Once again, I welcome all dignitaries and faculties, staff of the university, students and participants. Now, may I now request to Dr. S.D. Gorantiwar sir, Principal Investigator of CAST CSAWM project, Head Department of Agriculture Engineering, to give preamble about the project.
Thank you, Dr. Prashant. Eminent expert speaker, Honorable Dr. P.K. Gupta, who is honorary emeritus professor and INSA honorary scientist, Meerut University, Meerut, and at the same time, honorable chairman of today's international webinar, Dr. K.P. Vishwanatha, honorable vice chancellor of this university. Honorable chief guest and well-known person, Dr. K.V. Prabhu, who is chairperson, PPV FRA, Government of India, New Delhi, and respected Dean and Director of Instruction, Dr. L. Farande, respected Director of Research and Director of Extension Education, Dr. S.R. Gadar, all the associate deans, heads of the department, and all the participants and organizing team. Very good morning to all, and we welcome all of you for the first expert lecture of the international webinar series, which we are starting from today on this particular occasion. We have, in fact, very eminent personalities today who will be addressing all the participants. Around 680 joined through Zoom, and 100 plus joined through YouTube, and many more, they are still joining. And I would like to take this opportunity just to explain about this project in brief. This is the World Bank sponsored project under National Agricultural Higher Education Program of Indian Council of Agricultural Research, New Delhi, ICR. And the title of this project is Center for Advanced Agricultural Science and Technology for Climate Smart Agriculture and Water Management. And we have many more objectives, but important objectives of this project is to strengthen the academic program of this university, including MSc, MTech, PhD, for the climate smart agriculture, and also to start the postgraduate diploma, which we will be starting from this September. We have completed all the formalities. Second objective is to develop the technologies which are useful to the farmers, and based on, say, robotics, drones, Internet of Things, then other climate smart agriculture technologies, precision agriculture, and also to develop the climate smart agriculture villages and farms, which we have already started this, and we are in the process of completing this task as well. That is development of the climate smart villages, digital villages, and at the same time climate smart blocks. Our one important objective is the capacity building of the students, mostly PG students of this university, other university, young faculties for climate smart agriculture and water management. And we are performing this by organizing the series of training programs through offline and online mode under the guidance of our Honorable Vice Chancellor, Dr. K.P. Vishwanatha, Dean and Director of Research. And I hope we are also in this process train 10,000 plus participants and equal number of the farmers throughout India and few from abroad as well. So with this, I would like to remain because we have to listen to all these eminent personality. Thank you very much.
Now I would like to request Dr. P.L. Kulwar sir, Organizing Secretary, Member, CAST CSAWM project to give review of this webinar series. Sir.
Thank you very much, Prashant. Am I audible?
Yeah.
Yes, sir. Yes,
Good,
sir.
good.
So, respected Professor Gupta, who is the expert speaker for today, Dr. K.V. Prabhu, the chief guest of the today's program, Honorable Vice Chancellor, Dr. K.P. Vishwanatha, sir, all the dignitaries and the participants. Good morning to you all. As many of you know that today is the 198th birth anniversary of Gregor Mendel, the father of modern genetics. We decided to celebrate it by organizing a series of lectures on the theme of our project, that is climate smart agriculture, starting from today. Although few reports mention that the date of birth of Gregor Mendel was actually July 20, 1822, but several reports mention, including Encyclopedia Britannica, that July 22nd is the date of birth of Gregor Mendel, the day he was baptized. So we consider leaving that issue of July 20 or July 22, we consider July 22 for our program. That was also convenient for us also. And we decided to start this webinar series from today with expert lecture of Professor P.K. Gupta, who is a well-known geneticist in the country. As part of this webinar series, we are planning to organize expert lectures at regular intervals, and that will be circulated. Information of that will be made available in due course of time. For today's webinar, we received a huge response with 3,088 registrations from all across the country, as well as few registrations from abroad. This is in fact the highest number of registrations we received for any online training or webinar program for our project. And these participants include graduate and postgraduate students, teachers, researchers from India and abroad. Of these, 73% are the students, 23% are the faculty, and remaining are the participants from industry and other sectors. In terms of gender classification, 1,100 female candidates and almost 2,000 male candidates registered for this event. Just want to share for the information of the participant that Professor Gupta, even at the age of 84, is so active in research that people are so eager to listen to him, and therefore we got these many registrations to listen to his lectures. So with this, I would like to thank all the members for joining this event. And just for the information of the participant, because many participants always request for the certificate, so details will be provided to them in the chat box of the Zoom and the Facebook through which they have logged in. And at the end of the session, there will be announcement. So with this, I welcome again, and now I request Dr. A.L. Farande, Dean Faculty of Agriculture and Director of Instructions, for the welcome address and the introduction of the expert speaker and the chief guest. Over to you, sir.
Okay, can you hear me?
Yes.
Okay, fine. Thank you. Good morning to everyone. I extend my warm welcome to all. First of all, it is my privilege to extend warm welcome on behalf of Honorable Vice Chancellor Sir and all the faculties of our university to our beloved eminent speaker, Professor P.K. Gupta ji, Honorary Emeritus Professor and INSA Honorary Scientist, Chaudhary Charan Singh, University Meerut. I take this opportunity to introduce our eminent speaker, Professor P.K. Gupta ji, on this occasion of International Webinar Series coinciding with the 190th birth anniversary of father of modern genetics, Gregor J. Mendel. Professor Pushpendra Kumar Gupta sir started his teaching and research career at Meerut College, Meerut in 1958 and DAV College, Muzaffarnagar during 1959. He served as a lecturer in Gorakhpur University during 1960 to 69. He availed the Commonwealth Fellowship in Canada from 1964 to 1967 and did his PhD in wheat cytogenetics at University of Manitoba. He joined Meerut University in 1969 and continued for the remaining part of his life. He served in different capacities as a reader and then professor and head department of agriculture botany, now genetics and plant breeding, and dean faculty of agriculture till his retirement in 1996. He has traveled extensively throughout the world for various academic and research activities. After his retirement, he continued working in the Meerut University, now Chaudhary Charan Singh University, Meerut, in various capacities as a emeritus professor of life, a senior scientist, INSA honorary scientist, NASI senior scientist. He's also served as an expert on various academic bodies and committees for funding agencies during his long academic career. Professor Gupta has successfully completed large number of research projects funded by the various agencies. He has guided more than 80 PhD students, and majority of them are established scientists in India and abroad. He has published more than 400 research papers in national and international reputed journals. He has published about 24 textbooks on genetics, biotechnology, cytogenetics, which are very popular among the student and faculties. He is a member of large number of scientific bodies in India and abroad, and is a fellow of all Indian national academies in the area of science and agriculture. He honored with many prestigious academic awards, including the Birbal Sahani Gold Medal of Indian Botanical Society. His research contribution is well recognized in the field of plant genetics, plant breeding, cytogenetics, biometrics, quantitative genetics, crop biotechnology, etc. Sir, thank you for joining us for this international webinar series as an expert, eminent speaker. Now, I also extend my warm welcome to our beloved chief guest of today's function, Dr. KV Prabhuji, Chairperson, Protection of Plant Varieties and Farmers' Right Authority, New Delhi. I'm very happy that he has accepted our invitation as a chief guest in spite of his busy schedule and joined with us for his online international webinar series coinciding with the 190th birth anniversary of the father modern genetics, Gregor J. Mendel. Now I take this opportunity to introduce our beloved chief guest, Dr. KV Prabhu. He started his professional career in 1986 in ICAR and served in a different capacities till 2018. He headed the division of genetics IRI from 2006 to 2013 till he took over as a joint director research IRI New Delhi on 8th October 2013. Dr. Prabhu sir is instrumental in establishing the National Phytotron facility at IRI. He played an important role on the issue of redefining basmati rice in India and also led a wheat and barley improvement program of IRI from 2007 to 2018. He has been an active contributor to the crop improvement activities, which led to the release of more than 35 crop varieties, among which 8 of basmati rice and 8 from the mustard, 17 of wheat and 2 of barley. He is recognized for developing the team of plant breeders in the country, many of whom are leaders now in their fields. He has been conferred with the awards like Jawaharlal Nehru Award, ICAR Recognition Award in 2008, Platinum Jubilee Award of Indian Science Congress Association, BP Pal Award, VS Mathur Memorial Award, Norman Borlaug Award in 2012, Rafi Ahmad Kidwai Award 2012, and Atal Ratna Award 2019. He has elected Fellow of National Academy of Agriculture Science in 2005. He has guided 4 MSc and 20 PhD students and published 105 research papers in standard journals of international impact in the subject of genetics and plant breeding. He assumed charge as a chairperson equivalent to the Secretary Government of India of Protection of Plant Varieties and Farmers' Right Authority, Ministry of Agriculture, Farmers' Welfare, Government of India, with effect from February 6, 2018. I'm very happy to communicate that he is also an expert member of our Agriculture Research Council of MPKV Rahuri. Thank you, sir, for joining as a chief guest for this international webinar series. I'm very happy to welcome our Honorable Vice Chancellor, sir, and chairperson of this international webinar series, Dr. KP Vishwanathan, who is a guiding force in organizing such online programs in series during the lockdown period and always inspiration for us. I welcome you, sir. I also welcome Dr. S.R. Gadak, Director of Research and Director of Extension Education, for his special presence at this occasion. I also welcome Dr. X.D. Gorantiwar, convener and PI of this project, CAS project, Dr. M.G. Shinde, co-convener, co-PI, Dr. P.L. Kulwal, organizing secretary of all and all program coordinators and all my team cast members of MPKV Rahuri. I extend my warm welcome to all my officers, associate deans, heads of the department, faculties, and MPKV Rahuri. I also extend my warm welcome to all my dear participants within the country and abroad, that is from USA, Australia, Germany, Nepal, and Japan, for showing their huge response and active participation for this program. I congratulate to the convener, co-convener, and organizing secretary and course coordinators, all my CAST team members, for organizing this such a very important international webinar series at an important golden moment. But particularly celebration of this 190th birth anniversary of father of modern genetics, that is Gregor J. Mendel. Once again, I welcome you all and thank you very much.
Thank you very much, sir, for your welcome address. May I now request Professor P.K. Gupta to start his presentation, please. Over to you. Yes, sir, you can start.
Yes. Yeah.
Okay, Pawan. It's a great pleasure for me to be with you for the first time to this university. I'm very pleased to accept this invitation received from Dr. Palwal Kulwal, who has been my very dear student and one of the best students I ever had.
Oh.
And I also like to thank all the dignitaries of this particular conference, including the Vice Chancellor, Dr. K.P. Vishwanath, and also Dr. K.V. Prabhu from IRI has been very good friend. You will see that we had some collaborative projects with Dr. K.V. Prabhu. I will briefly mention about those projects later on during the presentation. Before I make this presentation, I have a word of caution. Most of this presentation you will find will consist of biotechnology and molecular biology methods which are used for improvement of crops. But the word of caution is that these approaches will supplement and will never replace conventional plant breeding. I always, in some of my publications also, I have mentioned this, that conventional plant breeding will still have the major impact on plant breeding crop improvement programs. And these newer programs of biotechnology and molecular biology will only supplement these conventional programs. Another important thing which I want to talk about before I start the presentation is that most of the climate change we are witnessing today are anthropogenic in nature. They have been created because of human activities. After 1950, Dr. Swaminathan has been talking about this anthropogenic era, they call it. And during this anthropogenic era, it is due to human activities that we are facing many problems. And I don't think there is any solution to this human activities which are responsible for bringing about this large number of changes in the climate. The outline of the presentation, you will see I have 5 bullets here. The first is expected climate changes, drought, heat, and carbon dioxide, and their direct, indirect effects, and their positive and negative effects. Just a few slides in the beginning, about 8 or 10 slides, they will cover this particular aspect. And then the efforts made in developing climate resilient crops. This will be the second part of the lecture. And the third part is methods of breeding climate resilient crops. How can we breed these climate resilient crops utilizing the biotechnological approaches? And then a very brief discussion on artificial intelligence and machine learning for crop improvement, which is the area which will gain momentum in future. We are going to use this machine learning and artificial intelligence in a big way in future. I'm sure about it. And I am myself very keen to learn about it. One of my friends in USA, whom I wrote to give me details about these programs, told me that we have to develop collaborations with computer scientists, and it will be difficult for us to learn it ourselves. And therefore, we have been in collaboration with Indian Agricultural Statistical Research Institute. There are a few scientists there who have been helping us in our biometrical analysis, QTL analysis. And for this particular program also, artificial intelligence and machine learning, we look forward to the cooperation from this particular institute. And the third part, last part of the lecture, about 15-20 minutes, will be the work done at Merit University. You see, I gave a lecture on the same subject, climate resilient, at eqtl in February. Some of the participants who are joining, they might have heard that lecture, but that lecture has been substantially modified because I don't like to repeat the same lecture more than once. And therefore, I have substantially modified this lecture. And particularly at eqtl, I did not talk about the work done at Merit University. And you will see that about 15-20 minutes in the end of the lecture will be devoted to the work done at Merit University, which is relevant to the climate resilient crops. You see here, I have shown the extreme climate related events which have taken place during 1990 to 2016, about 25 years. You will see that the events which refer to extreme temperature, drought, and then floods, and also then storm, the number of events which have taken place every year have been rising. And particularly they have been alarming in the year 2006 and 2008, and then gone down, but they are rising again. And the total adverse events which have occurred, you can see there has been a continuous rise in these adverse events, asking for preparedness on the part of plant breeders to develop climate resilient crops. Looking to the temperature during 1880 to 2020, during these about 140 years, you can see that the rise in temperature during the first 70 years, starting from 1880 to 1950, has been only 0.2 degrees centigrade. And because there is less of human activities influencing the temperature. But from 1950 to 2016, 2020, you see that there is 1 degree rise, 5 times increase in temperature in comparison to the 70 years during 1880 to 1950. Therefore, it is obvious that these anthropogenic activities are responsible for major changes which are currently taking place in climate.
Gautam, you can move to next slide.
Yes. Carbon dioxide changes have also taken place during the last, you can see in this particular slide that during 800,000 years before 1950, there was a rise in carbon dioxide concentration of only about 50 parts per million ppm, 250 to 300 ppm. And from 1950 to the current level is about 400 ppm, which is 100 ppm increase during the last about 70 years. This is again because of the anthropogenic activities which we are witnessing. For millennia, atmospheric carbon dioxide had never been above this particular line. And during the last about 70 years, you can see there is 100 ppm rise in carbon dioxide concentration. Then climate change has direct and indirect effects on crop improvement. There are direct effects on the phenotype, morphology, including physiology and productivity. We have witnessed it. There is evidence that there are changes in productivity because of climate change. Then there are indirect effects which influence crop productivity, including soil fertility, water availability, heat and flood and drought, sea level, rise in pests and diseases. They are also responsible for loss in yield in majority of our crops. And socio-economic effects are, there will be food demand is changing, escalating costs, policy, trade, unequal distribution of resources. All of these direct and indirect and socio-economic effects are affecting the crop improvement programs, and therefore we have to take necessary actions. Positive and negative effects. There are positive effects due to increase in carbon dioxide, because increase in carbon dioxide may increase photosynthesis rate, and therefore there will be increase in yield because of increased photosynthesis. Higher temperature may provide accelerated maturation, increased productivity, and possibility of growing new crops. These are some of the positive effects which we can foresee, but this increase in carbon dioxide have indirect effects which have adverse effects on crop yield. Negative aspects include extreme heat, drought, freezing, floods, and increase in disease, pests, and weeds may damage the crop. Ozone toxicity may also influence the productivity in the crops. Climate resilient crops, why do we need them? And I also, before I proceed further, I should also make it clear that climate resilient crops do not mean climate tolerant or climate resistant crops. Resilience is different than tolerance or resistance. We should bear in mind, because sometimes we confuse between the two, and we confuse climate resilience with climate tolerance. Adverse drought tolerance does not mean drought resilient crops. By 2050, half of the world population will be undernourished, according to some estimates. By 2100, 40% land surface will need to adapt to climate change. And in each decade, agriculture output will fall by 2% and the demand will increase by 14%. Urgent action, therefore, is needed to provide resilient crops for food and nutrition. And then the next generation green revolution to achieve the following two sustainable development goals. In sustainable development goals also, Dr. Swaminathan also has talked about zero hunger. So there is sustainable development goal number 2, which is asking for zero hunger by 2030. I am doubtful whether we will be able to achieve it ever. And the goal number 30 is climate action, how to deal with the climate change. Decline in climate resilience of European wheats. This is a very important aspect, and I came across this paper of 2019 published in Proceedings National Academy of Sciences, which has evaluated whether in the past we have lost climate resilience in the crops. And they took the data from 1991 to 2014, about, you can see 13 years trials, which have been conducted utilizing the same cultivars. 991 cultivars were utilized, and they consisted of winter, spring, and durum wheats. And they were grown at 636 locations in 9 different countries across Europe. And these trials have indicated that there was a decline in climate resilience of European wheats, surprisingly. And therefore, not only we have to improve the climate resilience, we also have to check the decline in climate resilience, which has been regularly taking place during the last about 20 years. And following steps, therefore, are needed. Identify agroclimatic variables which will influence the climate resilience, response to variables, grouping responses, grouping cultivars, assess response diversity. These were utilized for the estimation of climate resilience, and it was proved beyond doubt that the wheat has undergone decline in climate resilience during the last about 15-20 years. What has been done so far in climate resilience? You must have seen that during the last more than 20-30 years, we have regularly been discussing about the improvement of crop climate resilience in the crops, and necessary steps are also being taken, and large number of varieties are also listed which are claimed to be crop resilient. For example, in 2010, there was a national mission for sustainable agriculture where strategies for meeting the challenge of climate change was discussed. There is a document from Department of Agriculture, Ministry of Agriculture, Government of India. And then in 2015, also there is a document, Climate Resilient Crop Varieties. It gives a list of varieties, and I find that there are a large number of varieties which are claimed to be climate resilient for sustainable food productivity under aberrant weather conditions. But whether or not all these varieties are really climate resilient and whether they will be useful to deal with the problem of climate resilience, I am rather doubtful. In 2002, 2011, and 2019, there were 3 international group meetings held at Pune, Dharwad, Palampur. One of these meetings at Dharwad, I was fortunate to attend. And at these international group meetings also, we discussed how crops can be improved and how climate resilient wheat crops, wheat varieties can be developed. Then you must be familiar about the program of Harvest Plus, which has been introduced for about last 15-20 years. There is activity in Harvest Plus, and Harvest Plus has produced some varieties, particularly in wheat and rice, which are particularly nutritionally efficient. For example, they are rich in zinc and iron, and therefore they deal with the problem of nutritional security under the program of Harvest Plus, which has joined with Global Alliance for Improved Nutrition. So this particular program is not for improving the yield, but mainly for improvement for biofortification or improvement of nutritional aspects of the crops. Then there is a Hedwig program. Dr. Balyan from my department attended the meeting in Germany in 2013-14. And this program on heat and drought wheat improvement consortium was established, which will continue up to next year. And they regularly keep on discussing the problems of this climate resilience crops, particularly in the case of wheat. Developing climate resilient crops, what should be done? One of the most important aspects of developing climate resilient crops is to study the genetics of climate resilience. I must emphasize that we are talking about climate resilience, not about the tolerance, which we have been discussing. There may be some overlap, but they are not the same. We have to identify genes to be used for climate resilience. We have to determine whether climate resilience is mono, oligo, or polygenic, and prepare a list of genes of individual crops which may be used for developing climate resilient cultivars. We already know that all these characters which deal with climate resilience, they are all polygenic and they are complex and they are multigenic characters and complex traits, and they interact with each other, and therefore the genetic architecture for climate resilience will be really complex. There is no doubt about it.
Yeah.
And I will come back to that again later on during the presentation. And as I told you repeatedly, we have to remember that climate resilience is not stress tolerance. Methods for breeding climate resilient crops, there are a number of methods which have been proposed and are being used for the last about 20-30 years. Marker assisted selection is being successfully used for the last at least 20 years. I've been myself involved in this particular work, and therefore there is no doubt that marker assisted as a supplement to conventional plant breeding is going to be a utility and will certainly be used regularly along with the conventional plant breeding during the next several decades. And this marker assisted selection also includes marker assisted recurrent selection, particularly when you have to deploy a large number of genes, say about 8 or 10 genes. Then you cannot have simple marker assisted selection. Then you will have to use marker assisted recurrent selection so that you can keep on selecting individual genes and then intercross in the segregating generations so that you can pyramid a large number of genes through this method of marker assisted recurrent selection. Then genomic selection is another very, very important area. This was proposed about 20 years ago in 2001 and 2002. And during these 20 years, we have been struggling to find out to what extent genomic selection will be useful for improvement of crops and for developing climate resilient crops. I remember when Dr. Pawan Pulwal was in Cornell, I asked him that he should find out whether we can use genomic selection in our own programs. And he informed me that we have to wait. We are not really ready for genomic selection. So genome editing is another very important area of research, which is already being successfully used in several cases where CRISPR-Cas9 is a very popular technique which has been used. But more importantly, in the recent years, during the last 4 years, starting in 2016, we have another technique which is an improvement over CRISPR-Cas9, which is a modification of CRISPR-Cas9. And this base editing, I will give 1 or 2 slides. I will show how this base editing is done, because in this case, you are improving upon CRISPR-Cas and you are replacing only single base, single SNP, for example. If we know which SNP is responsible for the trait, then you can target only that particular nucleotide and replace it by another nucleotide to bring about the desired change. Then transgenics will, of course, be developed, have been developed in the past also to deal with the problem of climate resilience. But because of the regulatory problems and the bottlenecks and politics and the government restrictions on growing transgenic crop, we do not really know when transgenic crops will be able to reach the farmer's field, particularly in our own country. Although in countries like USA and several other countries, transgenic crops are being grown in large scale, but India has to still see, except cotton, we don't have any transgenic crop which has been commercialized in our country. Use of microbes. This is another very, very important area where you can use microbes, which is sometimes described as extended genome. The plants have their own genome, and then they have the genomes of microbes which are inhabited within the plant. And therefore, not only the genes belonging to the crops, but also the genes which are present in the microbes living within the plants are responsible for change of characters and for breeding improvement crop plants. And therefore, you cannot ignore the microbes. And therefore, we have to isolate microbes, try to find out the genes of the microbes which are contributing to improvement in crop plants. Then another combination of 2 or more approaches. We can, the above 4 or 5 approaches, we can combine 2 or more approaches together, utilize them together in order to, to develop climate resilient crops. And I told you earlier also that artificial intelligence and machine learning, they are going to be the most important areas of research in future, when the data will be not manageable by normal computers, and you will have to, yeah.
Mohsin, what happened?
किसी और का स्क्रीन शेयरिंग आ रहा है.
Somebody shared the screen.
No, okay.
Okay.
Okay, sir, you can continue.
Yes. You see, this is a picture I have downloaded only recently, where we see the evolution in the methods and strategies for developing climate resilient crops. For nearly 10,000 years, we were improving the crops and we were domesticating the crops on the basis of phenotypic selection, and the plant breeders are all aware. Students of plant breeding are also aware about this. And then, starting in 1950 or 1940, we started using the approaches. We started using quantitative genetics. We started using experimental design, statistics, and harvesters for improvement of crop plants. And therefore, during the last about 60, 70 years, we have been seeing that this particular area has been continuously utilized by the plant breeders. And during the last about 15, 20 years, we have been talking about genomic selection. To some extent, this technique has already been utilized in plant breeding, and genomic selection in this particular slide includes marker-assisted breeding. It is marker-assisted breeding, but it is not marker-assisted selection, because in this case of genomic selection, we make use of all the markers which are mapped on the chromosomes. We don't use individual markers. And then we predict on the basis of whole genome how the plant will behave when we select on the basis of all the markers which are known. And then the future will be the designed genomes. The genomes will be designed on computers, and then we will develop the designed crops, breeding by design, positive nucleotides, editing physiology, and things like that. You see, looking to the number of publications during the last about 20 years, you can see starting from 1990 up to 2018, yearly number of publications has been continuously increasing. For biotic stress, in 2018, there were 500 papers published in 1 year. Similarly, for abiotic stress, 900 papers were published in 1 year, 2018. And these papers do not include all the papers. They particularly include the papers published which utilized interval mapping and genome-wide association studies. I am not giving details of interval mapping and genome-wide association studies in this presentation, assuming that majority of the listeners, they would already know about it. But those who do not know about it, I must tell them that during the last 20 years, starting in 1990, interval mapping and genome-wide association studies have been the popular approaches for the study of genetics of biotic stresses and abiotic stresses, along with other particular traits. And this technique, both these techniques are being utilized extensively, not only throughout the world, but also in India. There are a large number of projects. There are ongoing projects on interval mapping and genome-wide association studies. And it gives useful information, very useful information, to supplement the conventional plant breeding. This is another particular area which has not been addressed thoroughly in our own country. There are few studies from India where eqtl analysis has been conducted and genetical genomics has been used for molecular breeding. This paper was published in 2001 or 2002. I teach my students how eqtls can be identified. Eqtls are actually the regulatory sequences, promoter sequences, which regulate the genes which are involved in individual traits, for example. This technique of eqtl analysis is similar to the technique of interval mapping, where you record the data, the genotyping data, and your phenotyping data in eqtl analysis is the expression level, and these expression levels are used as a trait. You measure the level of expression of a particular gene and record the data about the expression level and utilize this particular data then for identification of eqtls, which is very, very important, because in majority of cases, we only identify the structural genes, and even if eqtls are there, their effect is measured only in terms of the expression of genes which they regulate, but we do not identify eqtls in majority of cases. This, of course, is an expensive approach. This is why this has not been extensively used in India. But I would recommend that eqtl analysis for majority of the traits which are relevant to climate resilient crops should be studied in majority of the major crops. Once we have done interval mapping and GWAS for identification of markers which are associated with important QTLs or genes which are relevant to climate resilience, what should we do now? Do we continue doing QTL interval mapping and GWAS, or we have opportunities where we can utilize the information available through interval mapping and GWAS and extend further? One of the approach which our students are following in our lab is the meta-QTL analysis and ortho-meta-QTL analysis, which give us more reliable and robust markers to be used in marker-assisted selection and molecular breeding. Another important area which I have already mentioned is identification of EP-QTL, protein QTL, metabolomic QTLs, and with the help of these approaches, once you do this, you can identify the QTL hotspots, which will be very, very useful for plant breeding. And we can also identify heterotic QTLs, particularly for crops where heterosis breeding is very important. In this connection, there is a paper by Yang et al. 2018, which looks very relevant, a statistical procedure for genome-wide detection of QTL hotspots using databases. One such QTL hotspot I remember was identified by Rajiv Vashne at CRISET for drought tolerance in chickpea, and he has utilized that particular hotspots in developing drought-tolerant chickpea varieties. And therefore, such hotspots, rather than identifying simply QTLs, we have to identify QTL hotspots where a large number of QTLs may be present together, linked together, and may be utilized together for improvement of crop plants. Association mapping in post-GWAS era, we have recently published a review. Pawan Kulwal was one of the authors in this particular review in Advances in Genetics, where we have described in detail what should be done in the post-GWAS era once we already have enough information through the technique of genome-wide association studies. We have to make use of multi-locus and multi-trait GWAS. There are not many studies which have been conducted. There are 1 or 2 studies from our lab. There may be a few studies from other labs also. But multi-locus, generally in GWAS, we identify a single locus at a time and a single marker and a single trait. But that approach is not really so useful. Therefore, we have to extend our GWAS studies to multi-locus and multi-trait analysis. And also, you see, in majority of GWAS studies, we always ignore and reject the rare variants because of a statistical reason. And therefore, it is very, very important that these rare variants are also studied, because majority of the characters, majority of the genes, which are very, very important, they will be rare, and therefore they will be eliminated from the study and will cause serious problem in not identifying the important. And there are techniques available like CWAS and RWAS, where then GWAS meta-analysis is another approach which should be examined. It has been used in human beings, but not. I have not seen many studies in case of plants, but I am asking some of my own students to conduct GWAS meta-analysis utilizing MTAs which have already been identified, and then find out new MTAs which have higher reproducibility and higher precision and higher power. Causal polymorphism can be identified. We have never cared both in interval mapping and association mapping. We never cared to find out the causal polymorphism. We have been interested only in identification of markers associated with QTLs influencing the traits, but we have never tried to find out causal polymorphism, which are very, very important if you want to use genome editing and base editing, where you will have to know which SNP is responsible for the variation in the trait. Large number of SNPs showing causal polymorphism have been identified in case of human beings influencing different diseases, but I have not seen many studies where causal polymorphism have been identified in case of plants. Similarly, candidate genes will have to be identified and validated through studies which are being discussed in literature. Every year there are a large number of papers which are appearing now where causal polymorphism and candidate genes are being identified following the studies of interval mapping and GWAS. Can we improve upon interval mapping and GWAS? Same thing, we can improve upon it. You see, in linkage mapping or interval mapping is also described as linkage mapping, because in this case you are using the linkage and recombination frequencies for identification of QTL. Therefore, you can take a number of population, number of RIL recombinant inbred populations and conduct joint linkage mapping, number of mapping populations. If you use number of mapping populations, you are increasing the genetic variability which you are scoring, and you will get genes which have not been identified through simple population, single population in interval mapping. Similarly, there is a technique like JLM, joint linkage association mapping, where we combine the attributes of linkage as well as LD, linkage disequilibrium. So you are combining interval mapping and association mapping in a particular technology like described as JLM, which also has not been extensively utilized, although techniques have been suggested and models have been proposed by statisticians, but plant breeders have not used JLM to a very great extent. There may be few papers, but not many papers. Then you have multi-parental populations which have to be used. Magic population is one particular example which has been developed in number of crops and are being utilized for study of genetics. Then NAM population, nested association mapping population, which was first proposed in 2005-2006 in case of maize, and now these populations are being developed in some other crops also, which also is an approach which can be considered to be equivalent to JLAM, joint linkage association mapping, because in nested association mapping, we make a cross between a base parent, which is a common parent, with a large number of different genotypes. Sometimes 20 or 30 genotypes are crossed with the same single common parent, and then you have so many recombinant inbred line populations which can be utilized for a study where both linkage and association mapping are utilized for identification of genes. Precision and high throughput genomics and phenomics is the key to the success. You see, in both interval mapping and particularly in case of association mapping, we find that there are a large number of false positives. Particularly in association mapping, one of my students, when she was conducting the study, she found about 30 MTAs, marker trait associations, but when she applied correction, she found only 1 or 2. So a large number of association which are detected utilizing association mapping, they are always false, and therefore you have to apply corrections. And when you apply corrections, you get large number of false negatives. So you have problem, you have to, a trade-off. Correction also is a trade-off, and therefore you have to develop approaches where we reduce the frequencies of false positives and false negatives in our association mapping studies. So precision has to be improved. In case of interval mapping and association mapping, we always tell our students that association mapping has high precision but low power, and interval mapping has high power and low precision. And therefore, we have to combine these two. But in addition to use new approaches like JLM, JLAM, Another aspect which needs attention is that we need to use high throughput genomics and high throughput phenomics. And high throughput genomics is being used now, but high throughput phenomics is not being used to a very large extent. We have achieved high throughput genomics, but yet to achieve high throughput phenomics at a large scale in different parts of the world. Some efforts in this direction are being made. Some efforts have been made at IRI also, and perhaps some other centers also in our country. But to what extent these facilities are being utilized to generating data of high throughput phenomics and then utilizing this data in machine learning and artificial intelligence, that is a question which has to be addressed. We also need to identify causal polymorphism and candidate genes, as I mentioned earlier, following each internal mapping at GWAS. The data of internal mapping at GWAS has to be utilized. You see, sometimes you may utilize the data of the experiments which have already been conducted and then follow it up to identify causal polymorphism and candidate genes. Some of my students in our lab are doing this at present. For example, you can do metaQTL analysis, then you may do metaQTL analysis, and then a candidate genes, and these candidate genes can then be validated through wet lab experiments like qRT-PCR. We have published a paper, as I told you earlier, with Pawan Kulwal, Association Mapping in Plants in Post-Genomic Era, where we have described what can be done in future after the GWAS has saturated. High throughput phenotyping, a few slides showing how throughput phenotyping is important. Phenomics, control conditions versus field. The phenomics data can be taken in the field as well as in the control conditions. And there are forward phenomics and reverse phenomics. In the forward phenomics, phenotypic data allow us to predict traits of selection on the basis of phenotype. You take the phenotype and on the basis of phenotype you predict and select the plants. And the reverse phenomics approach, it explains why a given genotype stands out in a given environment. And both these approaches can be used and are being used in some parts of the world. And then in order to achieve and collect the data, high throughput data on phenotypes, unmanned aerial vehicles are being used, aeroplanes and drones are being used, not only in many other countries, to some extent in India also. And there are a number of different equipments available for recording this data. Pheno drone, 1SL, LIDAR sensors, nano hypersac, pheno drones. Some of these I will show you next slides. High throughput spectral phenotyping. You see, high throughput phenotyping does not mean simply phenotyping at a high throughput, but you have to use the phenotyping in different regions of the spectrum. For example, you will have to use cameras which will identify the compounds which absorb lights in different ranges of the spectrum, 400 to 700 nanometer, which is the visible range, red, green, and blue light, which is the visible range, then 900 to 2500, which is near infrared and short wave infrared regions of the spectrum, and then the UV. In 2019, I have seen a paper that UV region has also been utilized for phenotyping because there are compounds within the plant which absorb light, maximum light in the UV range only. And therefore, all these different regions of the spectrum are being utilized for collecting data because different ranges of the spectrum give you different informations, and therefore data has to be collected starting from 400 nanometer to about, starting from 200 nanometer, the UV range, up to 2500 nanometer, giving fantastic information which will be useful in machine learning and artificial intelligence later on. And this phenotyping can be done under controlled conditions and can also be done under field conditions. Under field conditions, I remember at IR also, Dr. Prabhu developed a phenotyping facility under field conditions. I visited that facility, but I do not know to what extent this facility has been utilized to collect data. Ground-based platforms are being used, PhenoCart. BISA is using, one of our students went there and saw this PhenoCart, use of PhenoCart, and we are also trying to obtain a PhenoCart because it is not very expensive. And then there are aerial platforms, including drones, and unmanned aerial vehicles, which are being utilized. A large number of papers are being published every year where these phenotyping under field conditions are being utilized, where aerial platforms, including drones and unmanned aerial vehicles, are being used. They are attached with different types of cameras, which will give you important information. This is a controlled condition where red, green, and blue imaging is being done with the help of different lights and different cameras which are attached. And these pods, you can see, some of you may be familiar, they move around the carrier belt and data is being regularly recorded. This is a pheno cart where you have a kind of bicycle with which number of different types of cameras are attached, and you walk through the field with the help of this particular pheno cart, and this pheno cart will keep on recording data, which can be utilized for conducting genetic studies. These are some of the facilities where you can record the phenotypic data in the field, very near infrared and short wave infrared facilities, interspersed images for recording data on field crops, bushes and fruit trees. You can see that these vehicles will keep on moving and keep on recording data from a distance in the air, and this data will provide useful information which cannot be obtained manually. This is aeroplane and drone, unmanned aerial vehicles which are being utilized. I have seen several papers. CIMIT is also utilizing aeroplanes to collect data, and drones are being utilized. Drones are being utilized in India also, and Pawan tells me that they are also planning to use these drones for recording data on phenotypes at Rahuri. Artificial intelligence with genomics and phenomics for climate resilient crops. This picture belongs to University of Tuscia, which is about 100 kilometers from Rome in Italy, where Phenobotics laboratory is there. And this laboratory, you can see that this is a field, particular field, where data is being recorded by 3 different equipments. These are Phenodrone 1SL with Nano Hyperspec. This will record data in different ranges of the spectrum, red, blue, and green, and also near infrared. And then these sensors are there attached with this particular vehicle. And then there is Phenodrone 2T, which is present here, and it will record data utilizing thermal infrared and red, blue, green cameras, which are attached with this. And they are all, both of them are aerial vehicles. And then you have a third vehicle here, which is a ground penetrating radar. And this will record data on the root architecture. It will penetrate into the soil and record data with the help of spectral phenotyping about the roots. And then once you have the data, this data is to be used with genomics and artificial intelligence to accelerate plant breeding. And then multiomics, you see, you have once recorded the data on the phenotypes. Phenotype alone is not sufficient. Therefore, you collect the data from genomics, epigenomics, transcriptomics, proteomics, metabolomics, and phenomics. And all these different disciplines give enormous data, which has to be utilized through artificial intelligence and machine learning. And this leads to integrated discovery. I myself do not fully understand this particular slide, but you should know the purpose of showing this slide is that the data, we normally utilize data from phenomics at a time, multibolomics at one time, but never try to utilize the data, multinomics data, including genomics, epigenomics, transcriptomics, proteomics, metabolomics, and phenomics. Machine learning for plant breeding, new genomic selection algorithm using machine learning methods, explainable artificial intelligence. These are being developed in many parts of the world. These are being developed. I'm sure in the next 5 to 10 years, this will be extensively utilized in different parts of the world. And then there is a paper published on machine learning methods for crop yield prediction and climate change impact assessment in agriculture. You can see that they are already being used, and this is why impact of using these different approaches are being analyzed And this paper describes some of these results. Epigenetics and epigenomics is another very important area which has not been addressed at the plant breeding level, but studies are being conducted where we are trying to find out to what extent epigenetics and epigenomics influences the traits which are of importance for plant breeders. Machine learning for plant breeding? Oh no. This epigenetics approach has 3 or 4 different disciplines. DNA methylation can be studied. There are a number of approaches. In our own lab, we have used a number of methods for the study of DNA methylation. You have histone modifications, a large number of different types of histone modifications. 8 of them are listed here. I will not talk all of them, but I will talk about 1 or 2 of them. And then there are microRNAs, including these are non-coding RNAs, which are also playing an important role. The more work is being done, the more we find out that microRNAs are very important components of the genetic architecture of the plants, influencing the traits of our importance. Then more recently, a paper was published by Vikram Gill from Kansas State University. He delivered a lecture in our lab on extra chromosomal circular DNA, which is another component of genetics. Large number of plants have utilized extra chromosomal circular DNAs, although they have not been studied extensively to find out to what extent extra chromosomal circular DNA are related with the different traits of economic importance. Epigenetic modifications have already been shown to influence flowering time, fruit development, plant immunity, and abiotic stresses. Therefore, there is no doubt that the characters which are important for developing climate resilient crops, they are also partly modified because of epigenetic component. Then yes, you see this particular picture, I will not spend much time on this because this is more complicated. It will take a few minutes to explain. But the idea of giving this picture is that the methylation, cytosine is normally methylated, but the context of this methylated cytosine is important. Sometimes methylated cytosine will be present just next to guanine. Sometimes it is present next to Hg, and sometimes it is present next to HH. HH can be anything except G. So these 3 different contexts of methylated cytosine are important. They are important in plants, not so important in case of human beings. And therefore, in our study also, not only we have studied the extent of methylated cytosine, which is present within the genome, we have also studied the methylation context, which is equally important to find out to what extent methylated cytosine will play a role. And there are a number of softwares also available which are being examined, which are being utilized by our bioinformatics lab in order to find out these different aspects. Histone chromatin modeling is very, very important, which is influenced by a number of different types of histone modifications. There are at least dozen different types of histone modifications which can take place, but we will not mention all of them. Some of you may be familiar, but 2 important modifications which I will mention and which we have utilized in our own lab in order to find out their effect on leaf rust resistance are histone acetylation and histone methylation. Histone acetylation and histone methylation are 2 different histone modifications which we have ourselves examined in our own lab to find out their effect on leaf rust resistance. And histone ubiquitination is another very important modification which we have not studied but is being. And all these modifications influence chromatin remodeling, which is very, very important for expression of different genes. Genomic selection is another very important area, genome-wide prediction in plant improvement. There is a paper in Trends in Plant Science which reviews the progress in genomic selection, and it has been shown that genomic selection has been more useful in case of animal systems than in case of plants so far, but rapid progress is being made in case of plants also, where genomic selection will be utilized. These are the two different components of genomic selection. You have a training population and you have a breeding population. The training population has to be used in order to train a training population and in order to develop a prediction model which gives you maximum precision in finding out the breeding values of the markers which we are using. So breeding value, estimated breeding values are estimated utilizing the training population, but training population itself has to be repeatedly used, cyclic, you can see, and prediction model has to be trained to get the best prediction model. And this is being done at many places. I have seen a series of papers published by Summit also in this particular area. But once you have the training population, suitable training population, we will record the data on the markers, breeding values of the markers, and these breeding values will then be used in the breeding cycle. This is the breeding, actual plant breeding cycle, where you will use the genomic breeding values in order to identify superior plants, and these superior plants may again be crossed together, and then the cycle may continue. So you can see that this is in theory, this is a very practical method and very, very important method, because in this case, you are making use the information of the whole genome rather than just a few markers. But we are nowhere close to extensive use of this particular technology so far. I'm sure within the next 5-10 years, we will be able to make rapid progress in using genomic selection in plant breeding. Steps in genome editing. Genome editing is another approach, another technique where CRISPR-Cas is utilized. You have a gene and a target selection, 20 base pair upstream of PAM sequence. PAM sequence is a proto-spacer adjacent motif which is present in majority of genes, and this particular PAM sequence must be present 20 base pair upstream of the gene in which you want to target. And there are hardly about 10 to 20% genes which have this PAM sequence available. Therefore, you have a limitation. Because of the necessity of the occurrence of PAM sequence, you cannot really edit all the genes in which you are interested. Then you design a guide sequence, and this guide sequence should match the target sequence and should express itself so that it goes and binds at the target site of the gene which you want to edit. And then you assemble this guide sequence and the Cas9, which is a nuclease, together, and then transform the plant. And then this guide sequence goes and binds itself to the site which we want to edit, and Cas9 will nuclease the DNA and bring about the desired change in which we are interested. And then there is regeneration and screening of the edited site, selection of edited plants for climate resilient, gain and loss of function, and functional genomics for climate resilience. But as I told you earlier, this is an example of successful use of genome editing. CRISPR-Cas9 has been used in developing these colorless mushrooms and drought-tolerant vaccine maize by DuPont company. Then I have been arguing, and some of the scientists have been questioning, why should I argue in favor of base editing? But I came across this particular discipline of gene editing when it was first paper was published in 2016, and after 2016, September 2016, a large number of papers on base editing have been published, where extensive improvement has been made in the technology of base editing. By definition, base editing approach enables the conversion of one target base at the end at one time into another in programmable manner using deaminases without double-strand breaks. In case of CRISPR-Cas, we use double-strand breaks which have its own limitations, or a donor template that are used in genome editing. So there are some of the limitations of genome editing which also give off-site changes, which is another problem. This base editing is more precise and therefore is recommended, and is now already being utilized in China and Japan, but I have not seen anybody working on base editing in our own country. And if somebody has started utilizing base editing in our country, of course, I would like to know. DNA adenine, you see, initially in 2016, cytosine to guanine change was suggested because deaminases were available for change of cytosine into guanine, but we did not have deaminases for adenine, and therefore they were artificially developed, these enzymes, and then they were utilized in base editing of adenine into inosine. Programmable base editing of adenine to thymine to guanine to cytosine. You can see that single base changes can be made utilizing base editing. And this technique is being regularly improved. And in case of human beings, there is a company established by the discoverer of base editor who is having business, particularly in case of humans. But this technique was developed at Harvard, where the technique was really developed for human health care. But subsequently in China and Japan, large number of centers are already utilizing base editing for the purpose of plant breeding. This is rice base editors. Large number of rice base editors, dozens of them have been developed during the last 2 or 3 years in China, which are being utilized for crop improvement. This is an example in case of maize, where herbicide-resistant maize was developed through the base editing. And this paper was published in 2020, January. Then plant breeding goes microbial. This is a slide which I have taken from Trends in Plant Science, published in 2017, where you can use microbes. You can isolate microbial flora from the plant, in the roots and from the leaves, etc., and then try to identify which microbes are useful to the plant. Once you identify them, you can inoculate with the microbes, or you can isolate the genes from the microbes and utilize them. And you can see that different microbiota utilized with the plant give you different phenotypes. The phenotype is different depending upon the microbiota which you are using. I had actually given a full lecture on this, but the time does not allow me to go into the details of this. But microbial plant breeding is another very, very important area which will be utilized in the future. One of the examples of this is by Uma Shankar, and this is his lecture which prompted me to learn about this microbial plant breeding. Uma Shankar at Bangalore University of Agriculture Science, he has conducted some experiments where he isolated endophytes from the salt adapted paddy crop, pokai, rice, and then selected saline tolerant isolates from these microbial cultures and utilizes these saline tolerant isolates to evaluate selected endophytes for salinity in sensitive genotypes. And he found that microbes can be fruitfully utilized in order to develop salinity tolerance in case of rice plants. And I'm sure I had a number of slides which I deleted later on, where this particular approach of utilizing microbes for plant breeding are being utilized throughout the world. Now I will spend another 10-15 minutes on the projects which we are handling at Meerut University: abiotic and biotic stresses, genetics, molecular bases, epigenetics, marker-assisted selection, including in silico work for novel genes in wheat. All these areas are being covered by a number of students and postdocs in our lab. Particularly, I will restrict my discussion to abiotic stress, in which we have drought and heat as the major components, and biotic stress, where we are using all the three rusts, mainly leaf rust and spot blotch. The collaborators for this particular work, we cannot do this particular work alone, and therefore we have collaborators from Washington State University, Kansas State University, CIMMYT, Mexico, Indian Agricultural Research Institute, New Delhi, particularly KV Prabhu and his team. They helped us in utilizing their facilities in order to do the work on leaf rust. Then University of Delhi South Campus has been very, very helpful to us, because majority of cases, some of the sequencing work which we have done and also other work was done in New Delhi South Campus. Akhilesh Tyagi, Jitendra Khurana, and Saurabh Raghuvanshi. These are the three faculty members who helped us in developing and getting useful results. and then Indian Institute of Wheat and Barley Research at Karnal, Dr. G.P. Singh and his team, and Bhudev Singh Tyagi, our old student, Gyanendra Singh, they have been associating with us in a particular DBT project. And then Banaras Hindu University, Arun Joshi and Ramesh Chand and their students have forced us to initiate some work on a spot blotch in which we were earlier not interested. But during the 5 years, because of the initiative of BHU, we have devoted time on a spot blotch also. This is a slide of one of my students, Vijay Gehlot and Vandana Jaiswal. They are happily married now, and both of them did their PhD in our lab and published a series of papers together and independently on different areas. Vijay Gehlot, in his PhD work, which he later on published in PLOS One, utilized this population Excalibur and Kukri double haploid mapping population, which was developed by Peter Langridge in Adelaide, Australia. And this population was available at Indian Institute of Wheat Barley Research. We obtained this population and he grew this population at 4 different locations continuously for 3 years. And the data was recorded and published in this particular publication. And you can see one of this particular picture shows multi-trait analysis. Large number of traits are there. They are correlated traits, and therefore you have QTL sometimes, which is common for all the different traits. Not many studies in our country involve multi-trait and multi-locus analysis, but in many of our studies, we have utilized both multi-locus and multi-trait analysis, and a series of papers have been published in our lab. There is another paper by the same group, Vijay Gehlot and his wife, Vandana Jaiswal. GWAS drought tolerance, 320 spring wheats were used in this case. These are the Manhattan plots showing candidate genes for marker trait association identified using mass. These are the candidate genes which were identified and are recommended to be used in plant breeding. Then another paper published by the same Vijay Gehlot and myself is two-component system. Two-component system is a system in which we utilize histidine kinases and response regulators. The particular climate change is perceived by histidine kinase on the cell surface, and then this is transmitted, the message is transmitted, and then response regulator transmitted to response regulators, which respond and regulate different genes which deal with the problem of these different drought. In this particular case, the drought response. There are a large number, you can see there is a large number of different types of histidine kinases. There are these histidine phosphotransperases, which is not found in case of microbes, but is found in case of wheat. And the message is transmitted from histidine kinases to histidine phosphotransperases to response regulators, and then response regulators deal with. And this work was initially done in silico. Large number of genes were identified, and then some of the genes were selected, and their expression was studied using qRT-PCR, utilizing drought responsive, drought tolerant, and drought sensitive genotypes, where it was shown that these genes are really responsible for developing drought tolerance. Another recent study, this paper has only just been published in Plant Molecular Biology, meta-QTL analysis for drought in wheat. During the last 5 years, the QTLs which were reported, they were used for developing meta-QTLs, and 13 different meta-QTLs were identified, which are now being utilized for identification of ortho-meta-QTLs, so that we will have more reliable and robust markers to be used in plant breeding. Another study, very, very important paper published in Theoretical and Applied Genetics. This is a collaborative study conducted by Australian group, Peter Langridge and his team with us, and Vijay Gehlot again was associated with this, and they used data of mapping population, the same mapping population grown in different parts of the world, including Mexico, Australia, and Europe and India, and then utilize this data in order to conduct QTL analysis, ultimately leading to fine mapping of a particular QTL on the chromosome 1b. And this particular was fine mapped, and the marker associated with this particular QTL, which has been fine mapped, can be utilized in plant breeding. So this is a very fine study which took many years, about 10 years to complete, but ultimately this paper was published, I think, this year, early this year. Genetics of heat tolerance in wheat. This is also, we have a project, USAID project with Washington State University, USA. For this particular project, our colleague Dr. Kumar is the principal investigator, along with Dr. Balian and myself as co-investigators. And under this project, there are several studies which are being conducted. AGPase enzyme has been studied in this particular case, which is thermosensitive enzyme, and there is variation for thermosensitivity and thermostarch synthesis, and therefore this can be used for plant breeding. And we first published a review, Saripalli and Gautam sitting with me, followed by another paper which was published by a lady, Ritu Batra, along with Saripalli, Gautam Saripalli. And in this particular case, they examined this particular enzyme in a number of monocots and dicots and studied the evolutionary relationships and the utility of the different genes. There are a large number of genes, at least 8 or 10 genes of AGPase enzyme, which are present in wheat genome, and they are being utilized for different kinds of studies. One of the students, Hemant Kumar, is studying AGPase enzyme at the enzyme level at different developmental stages of the seed and trying to find out the expression of this particular gene, particularly in response to heat. Then another paper on heat tolerance is under preparation and will be soon submitted for publication. That is the Jivas for heat tolerance. This is a part of the PhD work of Dr. Gautam Saripalli. And then QTL internal mapping is also planned, where we are using a DH population which was developed at Washington State University. Saurav Kumar is doing this work, and marker assisted selection on the basis of QTLs already reported is being conducted with TAPS. So you can see that in addition to drought, we also are conducting studies on heat tolerance, although more details are not available and not many results have been published so far. Then we are also, for nutritional security, we have a project by DBT, and we already, one of our students, Jitendra Kumar, who is working at NABI now at present as a postdoc, he conducted a study on Jivas for zinc, iron, and carotenoids, utilizing the same population for conducting Jivas, and identified a large number of different QTLs on different chromosomes, which can be used for plant breeding. Then leaf rust is another important component of our study, and we are very happy about the progress we have made in this particular area. And I must mention, I am pleased that Dr. K.V. Prabhu is here, and he would be pleased to see the progress. He already knows about it. We are dealing with leaf rust and spot blotch, and they are two different systems, and we are dealing with both of them. Leaf rust, you can see, is controlled by R genes, which follow gene for gene relationship, so that the R gene needs an AVR gene on the part of the host, only then resistance will be available. And if AVR gene is not present in the host, in the pathogen, then the disease resistance will not be there. As against this gene for gene relationship, we found that there is inverse gene for gene relationship in case of spot blotch. Spot blotch has different systems. You have genetic system which exhibits gene for gene relationship. You have QTLs, quantitative disease resistance, and you have this particular system of sensitivity genes which exhibits inverse gene for gene relationship. There is a gene, sensitivity gene TSN1 present in the host, and this sensitivity gene is essential for the pathogen to cause the disease. Toxic gene is the gene responsible for causing the disease, but this particular disease gene, toxin gene, will not be able to cause the disease unless this sensitivity gene is present in the host. And this has been verified over and again. Toxin is a virulence gene in the pathogen, and TSN1 is the sensitivity gene in the host. And because this is just the inverse of this situation, where pathogen avirulent is necessary for resistance. In this case, it is the host TSN gene which is necessary for causing disease. Both of them have been studied by us and Nilu Jain from IRI. We studied two different genes, LR28 and LR48. And both plant material for both of them was supplied by Dr. K.V. Prabhu through his work, important work which he has undertaken. And in case of LR28, Dr. K.V. Prabhu developed a pair of NILs, HD2329, which is susceptible, and HD2329 plus LR28, which is susceptible, which is resistant. And then he also supplied us the material of CSP44, which is single plant selection from an Australian cultivar, where LR48 is present. In this particular case, we collected material at pre-adult stage, which is susceptible, and post-adult stage, which is resistant, and then compared. In both these cases, we conducted cDNA AFLP studies. One of our students did it, and identified differentially expressed transcripts. Both these studies have been published. Transcriptomics and then epigenetics, including DNA methylation, histone modification, and non-coding RNAs have been studied. This is cDNA AFLP bands, and these bands were extracted, and then they were sequenced at the University of Delhi South Campus. Akhilesh Tyagi, he helped us in this, Jiten Khurana, they helped us. And then QRT PCR was conducted for 13 differential transcripts. They are not the genes, we do not know which genes are there, but 13 differential transcripts, when studied, we found that in the resistant, at 96 hours after inoculation, in the resistant, these transcripts express. In the other cases, they do not express. It means these differential transcripts contain genes which respond to the drought, and therefore must be important in developing drought resistance. Another paper on LR48, similar work has been conducted. The cDNA AFLP was utilized and the bands were again extracted, and then their analysis was conducted. For example, in this particular case, you can have, this is seedling stage, this is adult stage, and this is another stage, functional integrative genomics. This paper was published in Canada, Lethbridge doing some postdoc work. Then transcriptomic studies were conducted for both of them. Similarly, Chanchal Sharma, she was a lady who worked with us for a few years and then moved to Kenya, where she is completing her PhD now. And you can see that the information was collected in pairs. Differential transcripts were identified between early resistant to early susceptible, early resistant to late resistant, and then early susceptible to late susceptible, and so on. So you can see that there are 5 different pairs in which differential transcripts were identified, and it was found that a large number of genes were upregulated and another set of genes were downregulated in both the cases. So it simply demonstrated that the response to this resistance is very, very complex. The genetics which is involved is very, very complex, and large number of genes are involved. Next slide will also show you the same thing with APR gene. Same type of study was conducted. This paper was published this year by Nilu Jain in Functional Plant Biology. Then more recently, chromatin immunoprecipitation was utilized in order to find out the HD2329, 2329 plus LR28. This material, a project funded by NASF, and studies were conducted once again. Methylation using MSAP and MIDIP, methylated DNA immunoprecipitation, and methylated sensitive amplified polymorphism was studied. And in this case also, we are not identifying the gene, we are identifying the transcripts, and then trying to find out to what extent methylation is playing a role in resistance. This is a picture of methylation sensitive amplified polymorphism, where we are using 2 pairs of enzymes. They are isoschizomers, and one is sensitive to methylation, the other is not sensitive, and therefore you find different patterns of bands involving these different genotypes. Histone acetylation was utilized by Chanchal Sharma. She studied 6 different genes and found that promoters P1, P2 in these 6 genes behave differently and responded differently when they are acetylated or not acetylated. These are the names of the 6 genes which she studied, and this paper was subsequently published in Molecular Genetics and Genomics. Another paper was published recently, Plant Molecular Biology, July 5th, 2020, where methylation H3K27 and H3K4Me3, these 2 modifications were studied, and this detailed paper was published, and this complex network, which must be playing a role in providing this, in the role of this methylation in disease resistance, has been presented in this particular paper. And in all cases, in both these studies on leaf rust resistance which we conducted, the major conclusion is that there are more genes which become activated in the susceptible phenotype than in the resistant phenotype. And therefore, for achieving resistance, large number of genes have to be silenced, which become active in the susceptible. This is one of the major conclusions which we made, and we identified number of genes which become active in the susceptible parent, and they are silenced in the resistant parent. Then another paper published by Batra recently, this is under review, Science Reporter, where she identified these set domain genes, which are actually histone methyl transferases. She identified more than 100 different genes, which are histone methyl transferases, and is studying the role of these genes in, again, disease resistance. Another paper on microRNA and long non-coding RNAs. The work was conducted at IRI by Neelu Jain and her team, and the paper is being reviewed in Physiological Molecular Plant Pathology. And she found that large number of microRNAs are there, and each microRNA has a large number of target genes which is controlled by each of these microRNAs. Then this is a paper which I really like because this is new to me. Sudhir from BHU, who is Arun Joshi and Ramesh Chand, the student, he conducted this work at BHU, where he identified the presence of this particular gene TSN1, which is a sensitivity gene in more than 200 genotypes, wheat genotypes, and found that they are present in about 40% of the genotypes, and therefore you can utilize this information in selecting wheat genotypes which do not carry this sensitivity gene so that it will be resistant. You can see the spot blotch disease here. Then another paper on sweet genes. This is a sweet gene. They are susceptibility genes which are sugar transporters, and they are responsible for causing plant diseases. These sweet genes are hijacked by the pathogen and are utilized for nutrition of the pathogen, so that these are called susceptibility genes. Sweet genes are susceptibility genes, and therefore their recessive alleles are the resistant genes. And in rice, there are a number of these susceptibility genes where their recessive alleles are utilized for developing resistant cultivars. Some recent review articles have been published. I didn't want to go into the details of these, and most of you know that I am very fond of writing review articles, particularly in area which I am working on, and therefore large number of review articles during the last about 5 to 10 years have been published on drought tolerance, then transcription factors for drought tolerance, AGPAs. We published a review, genetics of yield, abiotic, biotic stress, and biofortification in TAG, biofortification and bioavailability of zinc, iron and selenium. This is under review in TAG. Sweet genes published in Molecular Biology Reporters, non-coding RNAs published in Current Science, synthetic chromosomes published in Current Science, and base editing for human health and crop improvement also published in Current Science last year. In summary, major efforts are being made for development of climate resilient crops. Following are some newer approaches. Beyond QTL interval mapping, you have to go beyond interval mapping and GWAS in order to identify the causal polymorphisms, and also in order to identify the candidate genes. Then base editors should be utilized to alter single bases. Plant microbiomes will provide extended genome which can be utilized. Epigenetics should be utilized. Once you know what kind of epigenetic modifications have played a role, then epigenetics can also be utilized. And the work in all these areas is being carried out on heat, drought tolerance, leaf rust, and spot blotch. And all this work was possible because of a team of workers. You can see a number of these faculty members are working as principal investigators and co-investigators in our department. Besides myself and Dr. Balian, there is Professor P.K. Sharma, who is a professor, Professor Shailen Sharma, who is professor and head, Dr. Rahul Kumar, who is a reader, and Dr. Sachin Kumar, who is conducting the heat tolerance project of USAID. He is a lecturer in our department. And then there are a number of these workers. There are two postdocs, these two ladies, Ritu Batra and Kalpana. They are experts in bioinformatics. They have published a series of papers on bioinformatics. Both of them, Kalpana has particularly done bioinformatic analysis from chromatin immunoprecipitation and also methylation bisulfite sequencing. And then Gautam, Tinku, they are completing their thesis. Both of the Gautam is working on heat tolerance, Tinku is working on drought tolerance, and then Irfat is working on disease resistance. Raki is working on nitrogen phosphorus utilization. She's writing her thesis. Kuldeep is working again on disease resistance. Anuj is working on drought tolerance. Then Vivud is working in marker-assisted selection for heat tolerance. Saurabh is working on QTL analysis on heat tolerance. Sunita is working on long non-coding RNAs. Hemant is working on AGPAs. And Manoj is working on pre-harvest sprouting. And Sahdev Singh is working on, on pre-harvest sprouting, QTL analysis, spot blotch. With these words, I thank all the listeners who spent their precious time. It is a pleasure for me to deliver this lecture, and I would be pleased to take questions from the audience or from the listeners.
Thank you very much, sir, for your excellent presentation. As it is witnessed by all, almost we are running with full capacity of 1,000 participants on Zoom, plus more than 600 participants joined through YouTube and Facebook Live. So thanks again. So there are several questions in the chat box. May I now request our coordinator, Dr. Prashant Kaya, to just start with the questions. So because there are so many questions, so they are picking the questions based on the theme of the your presentation. So Prashant, over to you.
Sir. Sir, one of the question is actual meaning of resilience. What is the actual meaning of resilience?
You see, resilience is different than tolerance in the sense that tolerance means that in drought it will respond.
Hello.
Resilience means that it will not be affected by climate change. Climate change. You see, there may be drought, or there may not be drought, there may be other conditions. Climate resilience means that it will not be affected by climate change. But tolerance does not mean that. Climate resilient also means that it will be resilient to different changes in the climate, not only one of them. But when we develop drought tolerant or heat tolerant plants, then you are responding to only one component and you are dealing with only tolerance. But you see, sometimes drought tolerant crop may not do well under normal conditions. So you have to be careful. When you are developing tolerant crops, then you must ensure that under normal conditions, its yield should not go down. And therefore, it is very, very important. Climate resilient means that the climate change will not affect.
Okay, thank you, sir. Can phenomics identify rare phenotypes?
Yes, rare phenotypes, you see, some of our students are doing. You see, once you have conducted genome-wide association studies, then you should concentrate on those rare markers and rare variants which you have in your population. And then either develop biparental mapping population utilizing them, or you try to correlate between the markers and the phenotypes. One of our students prepared a list of these rare variants and then compared the genotype with the phenotype to find out to what extent genotype is related. These rare variants or rare alleles are related to the phenotype. So this is an exercise which is being undertaken in many parts of the world, because these rare variants or rare markers, in my opinion, are very, very important, because the desirable trait would be rare, the desirable gene would be rare, and they would be present in these rare markers or rare variants, and therefore you have to capitalize on them. And therefore, once you have completed your association mapping studies, you should then concentrate on rare variants and rare markers.
Thank you, sir. Any relevance of cytogenetic studies in climate resilient breeding?
Well, cytogenetics is important once you know the position of the gene on the chromosome, then this may help you in further studies. For example, microdissection you may use, or for example, fine mapping. Cytogenetics will help you in fine mapping. But there is no direct, I don't think there is a direct relevance of cytogenetics. But cytogenetics, knowledge of cytogenetics is necessary, I must tell you, because many of our studies were possible because I knew cytogenetics. Many of my colleagues elsewhere at other collaborative centers, they could not do the same work because they were not proficient in cytogenetics. So cytogenetics makes you more competent to deal with the problem because you know what is happening. But if you are not a cytogeneticist, you don't have the knowledge of cytogenetics, then you cannot really deal with the problem.
Sir, in relevance to this, any chromosomal changes found in climate resilient crops?
I have not gone through the literature, but I'm sure there must be studies, because you see, in many cases, coming back to the question of cytogenetics, there are many genes which have been transferred from wild species. There cytogenetics become very important. You see, you may transfer segments from wild species, particularly in case of wheat. There are many examples. For example, in diseases, there are many examples where genes from Agilops, agropyron, and CKL rye have been transferred to wheat, and they have been responsible for improvement of the crop. And similarly, I am sure that for drought, heat, etc. also, there must be genes which are present in the wild species, and they can be transferred by genetic manipulation, cytogenetic manipulations to the, particularly, you see, for example, Dr. Dhariwal has conducted cytogenetic studies where he identified a large number of genes in alien species which are responsible for biofortification, zinc and iron fortification. He identified the genes and identified specific positions and also the segments which are responsible for zinc and iron. And I must tell you that only Dhaliwal could do it because he is cytogenetics. He knows cytogenetics, but nobody else could do it, even outside India. Even outside India, nobody could conduct a study involving biofortification and the utilization of wild species. Wild species. Parveen Chuneja is another scientist who knows cytogenetics and has been utilizing wild species. So wheat is a crop where wild species have been extensively utilized, and cytogenetics has played a major role. My own PhD thesis was on chromosome substitution, rye chromosome substitutions into wheat, and that made me get interested in cytogenetics. So cytogenetics knowledge, you can see that in case of biofortification, in case of leaf rust, also in case of, for example, I have talked about extra chromosomal circular DNA. This extra chromosomal circular DNA was identified in a herbicide-resistant weed by Dr. Bikram Gill, and he could do it only because he knew cytogenetics. Otherwise, it would have never been possible.
Okay, sir. Okay, sir. How breeding varieties will cope up with the change in virulence behavior of pathogens?
Oh, you see, virulence of the pathogen is something which will keep on evolving, and you have to keep on fighting with it. So you can't say that once you develop any resistant variety, the pathogen is not going to sit quiet. It will keep on struggling, and more virulent races and pathotypes will develop. And this is, for a plant breeder, it is a continuous struggle between the host and the pathogen. This is what is gene population dynamics, where the host will develop resistance, the pathogen will develop virulence, and the struggle goes on.
Okay, sir. How long climate resilient crop will adapt to the new environment?
Oh, well, that is also a continuous process. I think climate resilience, you see, for example, ICR has given a list, maybe 100 varieties, climate resilient varieties, which they have listed. If you test them, to what extent they can really be described as climate resilient, I am not really sure. So these climate resilient varieties, also like disease resistant varieties, may have to be improved further every time. This is a continuous cycle. So you can't develop a climate resilient crop and sit quiet and depend upon it. You will have to keep on exercising.
Thank you, sir, for the answers. I know that there are so many questions, but we can take only few questions. So may I request all the participants, if they want further details, Professor Gupta is always very much eager to listen to those questions and respond to those. So you can send him questions on his email, pkgupta36@gmail.com. So with this, I would like to thank Professor Gupta again for his excellent lecture and the answers to the questions of the participant. May I now request the chief guest of today's program, Dr. K.V. Prabhu sir, for his guidance, please. Over to you,
Thank
sir.
you. Thank you, Pawan. Sir, Dr. Gupta sir, congratulations. And so it was so fine a feeling to be listening to you again, just like old times. And to be with you, to be associated with you has been a fantastic experience, which I have many times expressed to you that I got in touch with you, not when I was at my youth. I was almost on the, say, lag phase when I got in touch with you. And you are in a lock phase ever, I think. You're never going to get into a lag phase, which is very, very nice. Thank you so much, sir, for this. And listening to Dr. P.K. Gupta, if the Vice Chancellor is there, I would just like to remark, is like listening to a music concert, where you can spend hours listening to the different alaps, to different levels of tals, and the way the musician takes the crowd, takes the audience along with him, and all of you both, he will enjoy the delivery and the listeners enjoy the delivery together. So that's what happens when we listen to Dr. Gupta. Thank you very much, sir, for this wonderful presentation on a very difficult topic. And I'm going to appreciate you didn't get into the complications which would put off people for saying it's not their cup of tea, which was very nice. Otherwise, when you're talking about these latest technologies, it's not going to be that easy for people to appreciate who have not been exposed to it, but the sheer lack of opportunity, which is the commonplace in Indian system, which you have been able to overcome. And that is an exception, being in Meerut. That too, see, I must tell the audience here, as a chief guest today, for which I thank Dr. Vishwanatha, it does not matter whether you're in Rahuri, or whether you're in Timbuktu or Meerut. The gusto and the pressure to learn and to do it with what you have is something that is what is resilience to the system. So resilience to system is an example of resilience breeding to climate stress, which Dr. Gupta said. His biography doesn't show Howard or Carolina or any such, other than his Canadian stint and some German stint, you see Agra, you see Gorakhpur, you see Meerut in his career. And Gupta is such a well-known name in Indian agriculture, like nobody else's. He has more than 44,000 people have used his citing, have been citing him. 44,000 is something. I think 10 of us put together will not account for 44,000. Among my best of my, I am supposed to be good in my subject. If I take my contemporaries, 10 best of them, it won't come to this. That is something great. So resilience to the system is something we have to develop in wherever we are working with. And that is what Dr. Gupta has demonstrated. And it is a feast that we have people like him around at 84 age. I mean, that's fantastic. I mean, the resilience of dealing with the system, resilience dealing with your own biology, and resilience to listen to the changing world of genetics, such that it may almost feel that the past 10 years work which you are doing is now obsolete. It may almost come to that. So you have to be resilient to that too. And he has done it very, very well. I was very happy the way he answered why cytogenetics is required. Fantastic. That's the understanding one should have. For example, people miss this boat totally, especially when we are dealing with alien, say, genes, which have been transferred into adapted crops. You will always consider the simple Mendelian inheritance pattern to say it is a capital A or small a and small a capital A and two chromosomes missing in the resistant type and present in the susceptible type. So whatever things of this kind you always consider. But when you are transferring an alien gene, there is a null allele in the other one which has received the new gene in the heterozygous combination. It's not heterozygous, it is hemizygous. So this understanding is possible within this understanding the moment you make a cross only with a cytogeneticist. Others will take it as other heterozygous. You will listen to people talking heterozygous in such situation. These simple things are most crucial when you analyze the data of sequence from those. If you don't see, you will not be able to make out why this difference is there. And especially when you're dealing with the hotspots and multi-QTLs and multi-trait as a common QTLs, you get into major understanding problems of the data, interpretation of the data, if you don't understand cytogenetics. So I like the way he is, which means nothing is obsolete, which means the system should be adapted to like we adapt to system. I would qualify further in stating, see, the tolerance versus resilience. He put it well by saying it is not the same. Absolutely correct. Resilience will have tolerance necessarily. Tolerance will never have necessarily resilience. Okay, that's the way to put it. Like all proteins have nitrogen and all nitrogen will not be protein. Something like that. Okay, so that is what it is. Resilience requires tolerance without any exception when it comes to climate stress related. The tolerance need not necessarily be for the less of it or the high of it, like low temperature, high temperature, both are temperature tolerance. So these are the things which resilience allow, and it's a tall order today to deal with it. And I think we have to deal with that only. Resilience is a better way of dealing with things today with such fast changing climate factors. And it is always going to be a little bit more advantageous dealing with abiotic stress than with biotic stress, although the biotic stress breeding job is much more precise because of the gene for gene hypothesis where it works, or even this inverse hypothesis which spot blotch part said. There also it is relatively easier compared to the abiotic stress tolerance. But abiotic tolerance once bred, it will not fluctuate as much as the abiotic stress tolerant material will say fluctuate because of the evolution of new races, which does not happen in this. In the case of abiotic stress, evolution of races will not happen. There is not a third genome which is going to interfere, but there could be range of the phenotype expression that will alter your necessity to improve on a developed crop. But that is going to be a longer period, and that's enough because a variety is not expected to stay along as a variety for more than 7 to 8 to 10 years, max 10 to 15 years, nothing more than 15 years, other than something very specific which the farmers have liked. which is what is happening with some traditional varieties even today. So those, in such cases, if abiotic stress tolerance is put in, you still have a longer life viability for those crops. So that these are the things which have to be worked together in any system today. And another important factor which we must take from Dr. Gupta's presentation is the requirement to have collaborations. Never be an endophyte. You have to be coming out of the niche where you are if you want to be identified in the niche, if you want to be around, if you want to be doing what you're supposed to do best in that subject, you have to reach out, you have to get to people. It does not matter whether you're, see, when Dr. Gupta first spoke to me, I was a senior scientist and he was already a fellow of all the societies and all the academies and such a big shot. But when he talked to me, it was like somebody who is in need of something which he does not have and he would like to take it from me. So it does not make any difference who I am. More than important, what is required is what do I have which the other person did not have. So that is an exchange which is required for both of them to grow. And that's what our wheat group has now enriched itself by being linked with Dr. Gupta. I have lamented, I have left the stage when I could have done much better than, because I was basically like Dr. Gupta, as oriented basic scientist for plant breeding with an applied viewpoint, and had a keener interest in application of basic understanding without leaving basic understanding, because my papers have always been basic and the material has been varieties. I've been successfully able to do it. Again, in the same logic as Dr. Gupta, with simple compound microscope, I could do several things with the alien substitution lines, which I got from CIMMYT, and understand the genetics of those materials, converting them to physiological traits. And that is how we could understand simple things by simple compound microscope and simple leaf area meter, looking at the graphic analysis of leaf area meter to look at how best we can identify the resistance as covered by a lower area of pathogenesis in a leaf area. So it is a question of understanding it and using your system to get the best that can be done out of it. Then world will appreciate, because output will speak, the product will speak, and that is the way one should adapt. And adaptation is the point. See, it is so good. Dr. I'm sorry, sir, I'm referring to you every other time, every other sentence possibly, but this is a wonderful advantage the 1,000 people around should listen to, because you don't get Guptas, Pushpendra Guptas everywhere and every university. You will not get. It's a rare phenomena, like somebody asked about rare phenotype. This is a rare genotype and rare phenotype. We should appreciate it and make best out of it when it is possible. Kulwal is slowly getting lost in the crowd of research part, accepting the situation of not being able to do research defensively, which is not Pushpendra Gupta. I'm sorry, Pawan. He referred to you as one of the best students. Live up to it. That is more important. So get on to that system. Review is fine, but fundamental basic research, project development, linking with the same universities is fine. Linking with other universities, do that. I have told your Vice Chancellor also about you, that he should be made to do it. Otherwise, he may escape, and after Dr. Vishwanatha leaves, there will be nobody to tell you, because I will also be out of the scene after some time, and to be able to be interacting with it. So it is important that you are a resource, and that resource should not be lost. In a system, whether it is for climate resilience or for any other requirement for the farmer, it is most crucial that we make best of this resource available. That is what Meerut University's this small unit of Dr. Gupta has demonstrated. So much, it has become a habit that they must tackle any point which comes up as a logic which has something that the basic sciences has allowed to understand. You must exploit it, not leave it, take it to the logical extent as much as possible. There will be some road that is available to overcome that problem. That is what has come out from that unit, and that is because of this ability not to get bogged down by what we don't have. What we have and who has what. Understanding this and linking there are the things we have to do. That is what everyone, whether it is Rahuri or say whichever place, whichever small place one is in, it does not matter. I have experienced the same thing when working in Tuticorin, where there was nothing. I was just having a glass house, nothing more, and I could still feel myself not having time at all to work with. I was alone. I had no other responsibility. All the time from morning till night, I was only in the say small cytogenetics lab, microscopy lab, and greenhouse, glass house. And that was it. And we could do several things. And that work which I did in first 5 years and in Tuticorin, I could not reproduce in this heavy environment of Delhi. That's because of this understanding what is required to be done and going deeper into it, for which you need to concentrate. So that is what is important, and I'm very happy that Dr. Vishwanatha has been organizing such seminars, which should inspire many of those younger participants who are there, who may be lamenting that they don't have some of the contemporary students who are now in Kansas or any other place where so much of good output is coming out, that they lament they don't have this, therefore they should stay put and they don't have to do it, giving this as a reason. That should not be the case, because there are many things which we can do. Holding plants, dealing with them, talking to them gives you much more than not looking at the plant and doing only say E-based outcome of informatics alone. It is not enough. That won't do much till you deal with the plant, connect with the plants. And nobody is better than you equipped with the plants without any advancements, but plants in real form as they respond to climate, as they respond to a system of stress or whatever. You watch that, use that difference you see, and that is how you are able to become a good scientist. So do that, people will come to you. People will come to you for the material. For example, there are very good cases globally. For example, Dr. Khush, when he was in Navy, he was a globally sought after man for the materials he had, not for the best knowledge in the most advanced techniques of breeding he has had, more for the material. Same thing with Ravi Singh today in wheat, in cement. He is most sought after because of the materials he has had, because of the understanding on each of the material genotype he has had. That is what it is, recorded information on each genotype which he identifies, selects. These are the things we can do in small universities, small places, and become important for the material and for the understanding of the subject, and be associated with the advancements. And advancements will come in, obviously, because material is the basis, and that's on which everything develops, everything works. That is what also helped me to a large extent to be with Dr. Gupta's team. It is important. Those things are required to be maintained. And I, as a say, a chief guest remark point, I would like to end here by stating this: do not look at the shortcomings which you don't, what you don't have. Look at what you have and make the best deal out of it. That is where the secret lies, and be sincere about it. That's all. Thank you so much, sir, for this wonderful opportunity. And once again, I thank Dr. Gupta for giving this wonderful exposition, which many students should be able to make out best. And I look for Pawan to having a good research paper coming out from his lab in next 3 years. Thank you.
Thank you very much, sir, for your guidance and your overview of the presentation and guiding all the participants. Regarding my personal, your concern about me, I can assure you that I will come to the expectation, and because we also got the DVD project on WheatWheat this year, network project. So we expect that in coming years, we'll have something to show to you. Thanks for your concern, sir. Thank you very much. So with this, may I now request Chairman of the today's program, Honorable Vice Chancellor, to have his remarks, please.
Good afternoon, everyone. Most respected Professor P.K. Gupta sir, and Honorable Chief Guest of today's webinar, Dr. Prabhuji, Chairman PPVF and FRA, both my directors, Dr. Gadak, Dr. Parande, and the COST team of our university, Dr. Gorantiwar and all his members, Dr. Kulwal. I saw Dr. Rajiv Varshni, another student, best student of Professor Gupta in the participants. He has commented, maybe he is there, Dr. Rajiv Varshni. And all my colleagues from MPKV, I have seen Dr. Asal here and so many other breeders, and all participants from all over the country and abroad. I consider it's really a great tribute to the father of genetics today. That is coming together more than 2,000 people, listening to a great geneticist and a very, I think, young at mind, young at this one. When I was studying for my undergraduate in early 70s, around 45 years back, we used to refer Dr. P.K. Gupta's book and for our UG, this one, and B.D. Singh, P.K. Gupta, and those books we used to refer. But when I saw him heard his first lecture, my first experience to listen to him was in 2002 in IRI. When I was attending, after 4 years of becoming professor, I became professor in 98 and 2002. I was attending because I belong to pre-biotech era. My PhD was in 1980s, 83. And I started attending, I attended 7 training programs in biotech and other things. That was my first listening to Professor Gupta. And later, till now, after 18 years, I have been listening to him 4 times. This 4th lecture I am listening. Each time I am just fascinated with his energy, with his enthusiasm. Hats off to you, sir. I think, I don't think anybody in the country, in the world, even 22 years after his retirement, he is having such an energy, such a zeal, and also such a, memory. It is not easy to pull all this data, and he was mentioning each and with each slide how he was explaining, and hats off to his memory. Sir, it's a great gift of God for you. I think we wish you will serve, and your teaching, our guidance will be available to our youngsters for many more years to come. And I pray God for your long and productive future in your this one. And coming back to this one, because we have already crossed stipulated time of 1 o'clock, I may not take much time, because as a customary, as a presidential or chairman's remarks, this one, I'm really overwhelmed with this one. Sir, the topic which you have covered, even Rajiv Varshni, he has put a comment. I read his comment at the bottom, because he was mentioning each time listening to new things comes out. And so your pooling of all these data, this one is really tremendous. And I think the youngsters, the type of questions our youngsters are responding, I think we have a great future. We need not worry about the future. I think our youngsters are their young brain are listening to this one. I saw almost 980 participants in Zoom, and also other more than 1,000 participants from YouTube. And sir, it's really a great tribute to Dr. Our Johann Mendel, who became Gregor John Mendel after he became the monk. This one, we know the history. Unfortunately, his work was not recognized during his lifetime, and it was rediscovered. I think all of us know the rediscovery. And this plant breeding is a science which started only in the 20th century. And it has become very fast. So I belong to the student of facet era. There was no calculator when we were studying this one. And we used to work with the facet. This one, I think Dr. Gupta may be knowing, and so many of our youngsters may not be knowing the facet which we are using for calculator. Our PG students from Bangalore and Dharwad used to book IRI calculation, this one, 1 or 2 months in advance. and they used to go travel 2.5 to 3 days to Delhi and do the calculation for their PhD and work. I know many of our seniors. That was the concern, and within short time, the science is developing fast. It's very difficult for me also to this one, and Dr. Gupta's the comprehension and his memory and this one. I'm thankful to you, sir, for accepting our request and our invitation to participate and guide the more than almost 1,000 to 2,000 students, youngsters to listening to you. And it's really this one, and we in not only teaching, not only writing books, your work in Meerut, your group you have shown and you have acknowledged each one of your young scientists. That is the thing. And I know Dr. Rajiv Varshni also since 2007 when I met in Mumbai and also association with ICRISAT. I know two of your students, one of our Dr. Kulwal and Dr. Rajiv Varshni, and all your students are very lucky to have you as a teacher. And we are all lucky to have this one. And I think the science of genetics or the science of crop improvement. We were beginning in 60s and 70s, we were breeding for yield. Then we shifted for the resistance, for biotic, now to abiotic. And sir, in Maharashtra, into almost 4 years back, when in one of the national seminar by ISGPB, we have decided to have a state level crop improvement program. Because of all the 4 vice chancellors, I was the only breeder. I suggested, instead of having each university, Parbani, Akola, Dapoli and this one, we have formulated a state level crop breeding program planned for resilience. Because we are taking the advantage of existence of NIASM in Baramati, that institute, and we are having the Dr. Is that what is the scientists from NIASM? They are providing very good support for our students. All our PG students are involved. We have developed a sort of long short breeding program involving, we are taking each university taking responsibility of important crops relevant to our area, and we have distributed and we have formulated a long short-term objective, medium-term objective and long-term objective of climate resilience, and we are working at it, and we need your guidance in formulating and executing this, and we may have to collaborate with some of the mission. Coming back to Kulwal, I think, sir, we with all these we have a very good phytotron facility, and he is looking after the biotechnology, and this one is doing well. He is having a collaborative program with ICRISAT on chickpea and also on wheat. And in addition to that, our university is falling short of the scientists and also staff. We are having the teaching load also. Anyway, they are trying our level best. To conclude, the plant breeding in India will be not easy in future. So it will be the breeders have to struggle a lot. Unlike the breeders of 70s, 80s, and 90s, it has to different omics are coming, and I think it is challenging for the youngsters to take this one. I hope our youngsters will take with the guidance of this one. I hope I am thankful to Dr. Prabhu. His contributions in wheat and many other crops from IRI is commendable, and both of you were as a very visionary speaker and also as a chief guest. I am thankful to both of you for guiding our youngsters and also our colleagues in our university without taking much time. Once again, I thank both of you, and I congratulate and compliment all the participants to make use of this one, and it's a great tribute to the father of genetics. Thank you very much, sir. Thank you.
Thank you very much, sir, for your chairman's remarks. So with this, may I now request Dr. S.R. Gadak, Director of Research of MPQ Rahuri, to kindly propose the vote of thanks, please. Over to you, sir.
Mr. Gadak? Okay, I think Kulwal, you only conclude because time is
Okay.
coming out.
So thank you very much, sir. May I now let me first thank Professor P.K. Gupta for his very elaborative and very extensive presentation, which was witnessed by almost 2,000 people from different parts of the country as well as abroad. So on behalf of the university, on behalf of the CAST project, I would like to thank him for sparing his time and accepting our invitation and for delivering this very wonderful lecture. Thank you very much, sir. I would also like to thank our chief guest of today's program, Dr. K.V. Prabhu.
Gadak Samage.
Okay, we request Dr. S.R. Garag, Director of Research, to please continue with the OTAP.
Yeah, yes, sir. Good afternoon to all of you, sir. Today I have got opportunity for making the OTAP thanks of this very important session. And the Center of Advanced Agriculture Science and Technology on Climate Smart Agriculture and Water Management, Mahatma Phule Viswavidyapit Rahuri, has organized 33 different events, including online training program and webinar on behalf of student and faculty member and farmers during the lockdown period. I call thanks to them. And today was the 38th program in the series, which was organized in coinciding with the 198th birth anniversary day of the father of modern genetics, George J. Mendel. I would like to thanks Honorable Professor K.P. Gupta sir, Emeritus Professor Chaudhary Charan Singh University, Meerut, for delivering an excellent lecture on the topic of climate resistance crop for the food and nutrition security. And importance of this topic is seen from the fact that the more than 300 to more than 200 people registered for this event. Secondly, I thanks to Honorable Dr. K.P. Prabhu sir, Chairperson of Protection of Plant Varieties, the Farmers' Rights Authority, Ministry of Agriculture, Farmers' Welfare, Government of India, for accepting our invitation as a chief guest in this occasion and guiding us. Thank you, sir, for your guidance. And I thanks to our Honorable Vice Chancellor and Chairperson, Chairman of this today's function, Dr. K.P. Viswanath sir. He is always guiding us, and he has guided from time to time to us. And thanks to sir for chairing of this session. I thanks to our colleagues, Dr. Ashokji Parande, Dean Faculty of Agriculture, and participants from different part of the country, as well as those who participated from the abroad, and also for sharing their views. And I thanks to the entire team of CAST team of MPQ Rahuri for their efforts for lockdown situation. They organized this program. I thanks to all of you. Thank you very much.
Thank you very much, sir. I think the program is now over. With the permission of the chair, can we close the session, sir?
Yeah. We will thank at the end both the speaker as well as the chief guest. Thank you very much, sir. Thank you. Thanks.
Bye-bye.
Thank you, sir.
Bye.
Thank you very much to all. Please end the session. Thank you.